Welcome to class, everyone. This is cell biology, and today we are going to be talking about cancer cells. We are in chapter 26. So, as just some introductory background information, cancer is a disease that arises from abnormalities of cell function. It's not an infectious, contagious disease. It is a disease that is something in your cell is not functioning correctly. And when that happens, you'll have some cells dividing in ways that they shouldn't be. They'll be accumulating in uncontrolled ways, creating in a mass of cells that form a tissue called a tumor or a neoplasm. So, one of the underlying crucial issues here is not that the cells are dividing faster. It still is going to take a cancer cell about 24 hours to go through that cell cycle, just like it takes a healthy cell. The difference here is that cell division has become untethered to cell differentiation and cell death. So cells are continuing to divide without appropriately differentiating, turning into the proper function that they should have, without dying at the time that they should. And so you end up with this mass of tissues. Underlying all of this are changes in your genes. It can be gene mutations, or it could be changes in how genes are expressed. It could be even something epigenetic and temporary, or it could be a gene that is multiplied and you are having an increase in gene expression. That is what's going to cause cancer. Cancers can be divided into two different kinds of tumors: a benign tumor and a malignant tumor. Benign tumors are tumors that stay in one local area. They're confined to one space. So because of that, they're not as dangerous as malignant tumors. They're not spreading to other cells, so they're not going to be impairing the function of other organs. They're also, because they are in one area, it's easier to remove them by surgery or by targeting them with radiation. Compare that to malignant tumor that is able to invade other tissues. It can start affecting other organs of your body, organs that are even far away from that original tumor. It can make it more difficult to treat because you don't just have one confined tumor to remove, you'll now have them throughout the body. And in general, cancer is going to refer to a malignancy, particularly malignant cells that can spread to new locations. Cancers can be grouped into three different categories depending on what kind of cell they arose from. About 90% of cancers are carcinomas. These are cells that arose from epithelial cells. Those epithelial cells could be covering the internal body surfaces or the external body surfaces. So this would be things like lung cancer, breast cancer, colon cancers, kind of the big ones in the United States. Sarcomas are going to develop from a lot of that tissues that arise from like the embryonic mesoderm, things like supporting tissues, bone, cartilage, fat, muscle. Lymphomas and leukemias both are cancers that arise from blood and lymphatic cells. The difference is lymphomas tend to grow into solid tumors, and leukemias tend to be cells in the bloodstream. So we mentioned one thing that causes cancer is that cells are not developing and differentiating how they should be. Cell differentiation is a way that a cell can acquire specialized properties. So something like a stomach cell gaining the ability to help function in the digestive system. So as the cells are differentiating, as they're gaining specialized traits, they're often losing the ability to divide. So differentiated cells are typically not the ones that are multiplying. It is typically stem cells underlying that are going to be able to form more cells. Stem cells can form more stem cells like itself, or stem cells can become more differentiated and specialized cells. So here's two examples. This is a healthy section of skin, and this is a section that has a tumor in it. So in this underlying tissue, you have this layer of stem cells that are dividing. But when the cell divides, this light brown cell divides, it has two daughter cells, one of which differentiates into a skin cell, and one of which stays as a stem cell. So you have one differentiated cell that's not dividing, one cell that is. This cell divides into two. One daughter cell becomes a skin cell that's differentiated, one stays a stem cell. So you have this process of cell division going on, but you're not creating too many cells. As the cells are creating more skin cells, they are moving on up. Eventually, your skin cells are dying and just being shed. Compare this to this tumor here, where you have this kind of messed up kind of stem cell. And as it divides, it might divide into two cells, one that's supposed to be differentiating, kind of, and one that's kind of still a messed up stem cell. But then this one divides into two stem cells. So suddenly now both of these are dividing into cells. Then this one is going to divide into two stem cells. So you have this accumulation of these inappropriately behaving stem cells-ish that are dividing and accumulating in a way that it shouldn't be. They're not differentiating into healthy skin cells. They're just growing into this mass of cells that are dividing. It's not that it's dividing at a faster rate. It's still taking just as much length of an arrow, just as much time to grow, but you end up with all of these cells instead of just these. You're continually going to have this tumor growing because new cells are just being produced in greater numbers than you need them to, in greater numbers than your body can support and sustain in a healthy way. In our cancer cells, we're going to see some particular traits that give them the ability to form tumors. And we can study a lot of these cancer cells by experiments with nude mice. Nude mice don't have an immune system, and so if you inject cells into them, you can see how they're growing. A normal human cell, if it's injected into a nude mice, it's going to give itself the signals that it shouldn't grow inappropriately. So normal human cells injected into nude mice don't grow. Cancer cells, however, don't have those signals to stop growing. So those cancer cells injected into a nude mouse will keep growing, keep proliferating, and form tumors. One change we see between these cancerous cells and the normal cells is anchorage dependence. So normal cells are dependent on anchorage. They're dependent on being attached to some sort of surface. This is whether they're in culture or even in your own body. Cells grow best when they're attached to some sort of surface. Cancer cells have lost this requirement, so they're okay to grow when they are attached to a surface. They're also okay to detach and spread throughout the liquid, spread throughout your body, and keep growing. So those cancer cells, because they are not dependent on anchorage, they can grow without being anchored to something. Cancer cells are said to be anchorage independent. In your body, normal cells are going to meet that requirement for anchorage because they have these proteins called integrins. Integrins are proteins on the surface, the exterior surface of the cell, that allow them to attach to the extracellular matrix. That gives them the signal that they are attached and they can start growing. You can block this attachment to the matrix, and in that case, if a cell is not attached, a normal cell is going to give itself the signal, uh-oh, I'm not doing something right, and it'll self-destruct. The way a cell self-destructs is through apoptosis. A cancer cell that is not attached, it doesn't wait for that integrin to say it's attached. It is fine to keep growing. It doesn't have this whole safeguard. It doesn't die when it is unattached. It just keeps growing. Another thing that cells do is they grow until they have a single layer of cells. So you may have noticed, like if you cut yourself, your cells will grow to heal that wound, but then they'll stop growing after that. It doesn't keep growing after you've healed. In a lab setting, you might have like a plate covered with cells. And if you scraped those cells, you have simulated a wound. The cells will grow back to fill in that surface, and then they stop growing. Once they have a single layer of cells, they stop. That monolayer stage is as far as the cells are going to grow if they're normal. That is called density-dependent inhibition of growth. So density-dependent, they're able to tell they are in contact with cells on all of their sides. And when they have that density dependence, when they've realized there are cells all around them, that inhibits their growth. They stop growing. Cancer cells don't care. They just keep growing. They'll pile up layers and layers of a cancer cell. They don't have any sensitivity to density dependent inhibition. Another thing that cancer cells do to keep them growing is they have found a way to keep their telomeres long. So we first started studying this in HeLa cells. HeLa cells, this word he comes from Henrietta and la comes from Lacks. Henrietta Lacks was a patient with uterine cervical cancer that were harvested from her grown in cell culture in 1951. Today I'm making this video in 2024. These cells are still being used in lab, growing well. A normal cell is not going to grow that long in culture. Normal cells have a certain length of time that they grow, and every time they divide, they're able to tell they're getting a little bit older, and eventually they stop growing. They develop some senescence and stop growing. But cancer cells don't have that limit. They will keep dividing indefinitely. What we found is that every time a cell replicates, its telomeres get a little bit shorter. Telomeres are sequences of DNA at the end of each chromosome, and we're not very good at replicating our chromosomes, so every time your cell divides, those ends of telomeres get shorter and shorter. So as a cell ages, as an organism ages, their telomeres get shorter and shorter and shorter. And eventually the cell will divide so many times that the telomeres are too short to protect those ends of the chromosomes. That triggers apoptosis, it triggers cell death. And that's it for that cell. Most cancer cells produce an enzyme called telomerase. Telomerase is an enzyme that is active in embryonic cells, so that babies are born with full-length telomeres, with brand new young acting cells. But as adults, our telomerase should not be active anymore. Cancer cells activate that telomerase. That telomerase is going to keep those telomeres nice and young, and keep those old cancer cells thinking they are young spring chicks that need to divide. So by keeping long telomeres, those cancer cells keep growing. They cannot tell that they have divided enough times, they're old enough, that's it for them. They just keep growing. Okay. Those mechanisms to keep their telomeres long allow cancer cells to keep dividing, but they're not what caused the cancer to divide to begin with. It doesn't stop it from dividing, but it doesn't cause it to divide. Does that make sense? So cells are going to also need a growth factor. So in our next video, we're going to come back and talk about some different growth factors involved in cell division. We're going to talk about some different ways that cells avoid their cell cycle controls, different ways that they avoid apoptosis, and ways that they're able to keep their tumor growing. All right. I'm glad to meet you this semester. I'm excited to spend this class with you, and I'll see you in our next video. Bye.